Okay, first things first. Let's get one thing straight. Cells are not just these simple static blobs you might remember from a textbook. They are unbelievably diverse. I mean, just look at this. This is a classic example. You've got this hunter neutrophil, that's one of your immune cells, literally chasing down bacteria. And all around it, you have these passive red blood cells just kind of floating along. Right away, this tells you everything. Cells have totally different shapes, sizes, and, well, personalities. And this diversity, it absolutely extends to their size. I mean, the scale is just staggering. It spans orders of magnitude. You've got a tiny bacterium, which is just a few microns long. Then you've got a human egg, which is actually big enough to see without a microscope. And then you have an ostrich egg. That giant thing is a single cell you can literally hold in your hand. Wild, right? So even with all this crazy variety, it turns out that all life on Earth is built from just two basic blueprints. And this split, this division, it's one of the most fundamental forks in the road in the entire history of life.
On one side, you've got the simple prokaryotes. Think bacteria. Their genetic material, their DNA, is just kind of floating around in the cytoplasm. But then, on the other side, you have the complex eukaryotes. Now, these are the cells that make up you, me, every plant, every animal. And their defining feature? Compartments. They're full of these little membrane-bound rooms, with the most famous one, of course, being the nucleus.
Okay, so what is it that creates these super important compartments? Well, to get that, we need to zoom in on what is maybe the single most fundamental structure of the cell, its outer board.
This barrier has a name. It's called the lipid bilayer. And you know, I love how MIT professor Adam Martin puts it. He says understanding the structure is so freaking important. And he's right, because this barrier is literally what makes a cell, well, a cell. And how it forms is just a marvel of physics, really. It's all about self-assembly. So each of these little lipid molecules has a head that loves water and a tail that hates it. So what happens when you throw them in water? Well, they automatically, spontaneously, arrange themselves into this two-layered sheet. The water-loving heads face out, and the water-fearing tails all hide in the middle. And boom, you've created this oily, hydrophobic core that most molecules just can't get through.
And the result of that simple barrier is, well, it's profound. It means the cell can create a totally different world on the inside compared to the outside. I mean, just look at these numbers for different ions. There's like 10 times more sodium on the outside, but over 30 times more potassium on the inside. This is not balance. This isn't some placid equilibrium. This is a state of tension actively maintained with energy. You could say this is the very definition of being alive.
All right, so if this membrane is such an amazing barrier, that raises a question, right? How does a cell get big stuff in or out? I mean, these things can't just squeeze through. The cell needs like a dedicated shipping and receiving department.
So to bring things in, the cell uses this incredible process called endocytosis. The membrane itself isn't rigid, it's dynamic, it's fluid. It can literally fold inward, wrap itself around a particle, maybe it's a piece of food or, even a virus, and then it pinches off, basically swallowing it whole into the cell. And the particle ends up inside this little bubble called a vesicle. And to send things out? Well, it basically just runs that whole process in reverse. It's called exocytosis. You've got an internal vesicle, it's loaded up with cargo, maybe some hormones or neurotransmitters. It travels to the edge of the cell, fuses with that outer membrane, and just spits its contents out into the world. It's literally the cell's postal service.
Now, this whole process of engulfing stuff, it's actually tied to a totally fascinating, mind-bending story about where our own complex cells came from. So let's journey a little deeper inside, into the world of organelles.
And we're going to focus on one in particular, the mitochondrion. Now, I know what you're thinking. The powerhouse of the cell, right? Everybody learns that. Well, that's a massive, massive oversimplification. In reality, mitochondria are these dynamic, constantly moving, tube-like networks that can stretch across the entire cell. They're not just little bean-shaped power plants. And their complexity, it just begs a huge evolutionary question. How in the world did our ancient ancestors, those very first complex cells, get such intricate, complicated machinery on the inside? Well, the leading explanation is a theory that sounds like science fiction. It's called the endosymbiont theory. And it basically says that billions of years ago, some big ancestral cell engulfed a smaller bacterium, but instead of digesting it, it just kept it. They formed a partnership, a symbiosis, a cell living inside another cell. And what makes this theory so powerful isn't just that it's a cool story. It's the evidence. Get this. Mitochondria have their own DNA, and it's a little circle, just like bacterial DNA. They reproduce by splitting in two, just like bacteria. And this is the kicker. Our cells can't make new ones from scratch. You have to inherit them. They are, for all intents and purposes, ancient organisms living as tenants inside every one of our cells. Okay, for our final stop on this journey, let's look at the machinery that gives the cell its shape, that allows it to move, and that performs these incredible feats of mechanical strength. Let's talk about the cytoskeleton.
Now, when you hear the word skeleton, you probably think of something static, like our bones. Forget that idea entirely. The cytoskeleton is the opposite of static. It's a dynamic network of protein filaments that are constantly growing, shrinking, assembling, and disassembling. They're like little molecular machines, tiny motors that generate actual physical force.
So, a key player in this whole network is something called a microtubule. Think of it as a hollow, tube-like polymer. And its superpower is that it can grow and shrink incredibly fast. And by doing that, just by growing and shrinking, it can generate some serious pushing and pulling forces inside the cell.
And we are talking about immense force here. In lab experiments, a single tiny shrinking microtubule can generate enough force to literally pull a glass bead through viscous liquid. That's more than enough power for what is probably its most critical job of all, moving our chromosomes.
So here's where it all comes together. When a cell gets ready to divide, these microtubules build this incredible machine called the mitotic spindle. They grow out from opposite ends of the cell, they attach to the chromosomes that are all lined up in the middle, and then they shrink. They start pulling. And this powerful physical pulling action is what separates our genetic blueprint, making sure that each new daughter cell gets a complete and perfect copy. It's literally mechanical engineering at the microscopic level. So think about what we've seen. From that smart, self-assembling outer membrane, to the ancient bacteria living inside as organelles, to the molecular motors that literally pull the cell's own DNA apart, it's clear the cell is not some simple blob. It's a bustling, microscopic city, a whole universe of dynamic machines. Which leaves you with one final thought. If just one of these cells is its own universe, what does that say about the trillions of them working in concert right now to make you?